wanted to talk today about a video that I've been shown. It's in BitChute or also Truth Media, and it's called Covid-19 is a parasitic artificial intelligence nano-weapon, which is a bit of a mouthful, and it shows us some interesting techniques that misinformation people use or people accidentally use to sell us incorrect pieces of information or correct pieces of information badly. One of the things that they do, this is Karen Kingston, she's talking to the moderator, who's this guy with the rather mean-looking face there, and they discuss corona and the vaccinations, and they throw out a whole lot of things. So the techniques that they use are the Gish Gallop, which is where you vomit out a huge amount of information very, very quickly, very often low quality information, easily debunked information, but because of the amount of it, it becomes very difficult to keep up. As a viewer, obviously, online videos have the disadvantage for this that you can stop them and look up what they're talking about, but still, if you ever go and look at it, I won't show very much of it on here because of copyright and all kinds of things, but I won't show very much on it either because it is bad to copy somebody's bad information, to reiterate it, because I'm trying to debunk it. The Gish Gallop has the advantage to the person who does it that he will always be left with little bits left over that the opponents won't be able to resolve, or in the case of this online version, it provides so much for anybody to do to debunk it all that it is very difficult to catch all of it. The next thing that she does is what's called quote mining. She goes through papers in this case, finds small comments and shows them, and that they appear to support her point. The whole paper doesn't necessarily support her point. We will come to an example later. That is very commonly used tactic of partly sometimes of religious people, and sometimes in religious arguments when everybody gets very upset and excited, and also in many other arguments, political arguments. For example, Ben Shapiro is well known to do this as well as the Gish Gallop. She also implies things that she doesn't specifically say are there, so she says something and then continues into a completely different theme as if it is connected when it isn't. And this is another type of bad practice. If you look at this video, you will see all of those things. Maybe in the future, when you are exposed to people telling you things, you will think about what they're doing to you. So what do we have here? First of all, she sets out and says, or actually the moderator says she can prove it. She says she can prove it, he says she can prove it. No proof is actually ever presented, or evidence, as people would prefer. Scientists would prefer you to call it evidence rather than proof, and no evidence is presented. Some very dodgy information is presented, but no evidence that what she's talking about, or that she even knows what she's talking about, is ever presented. She starts off saying Covid-19 is supposed to be mRNA, which isn't true. The vaccine is supposed to be mRNA. Covid-19, or coronaviruses in general, are sense RNA viruses, which means they also contain a type of mRNA, but it's not the same as mRNA. It comes out, it encodes for new virus particles, making new virus particles and making them infectious. So it is not that different from mRNA, which is a type of RNA. It's a type of nucleic material that cells use as a blueprint for making proteins. So effectively, DNA copies into messenger RNA, mRNA, which is then sent to the factories of the cells to make proteins, and whichever code is on the mRNA, you get different proteins. The idea of the mRNA vaccine is that it codes for a protein that the virus would otherwise make on its outside coat, the spike protein, and that is what you then become somewhat immune to. And then she gets into what could be very dubious territory, because she accuses a guy called Ralph Baric of inventing coronavirus.
Government documents and the inventor of SARS-CoV-2 himself, Ralph Baric, has clearly stated that all mRNA coronaviruses, including SARS-CoV-2, are impossible to achieve.
Which is completely untrue. He didn't do that at all. He worked on coronavirus before it jumped into humans and was looking at how likely it was to be able to do that. And he, at the time, said it would be very difficult for it to do that. She correctly notes that that is true, but she claims that he's claiming that it would have to be artificially manipulated to do that. He was looking at it in 2016 and saying that it's not going to jump right now, but it is a serious danger. She then goes on to correctly state that mRNA, or people show that mRNA falls apart very easily. It does that outside the body in water. It also does it outside the nucleus of the cell, because outside the nucleus of the cell there are enzymes that are specifically there to break down RNA. So it doesn't last very long, and it is not very effective at getting into cells, because typically mRNA should be made by the cell itself and transported to the place where it's going to be used. Of course, RNA is difficult to stabilize. That's why it is done typically in some kind of structures and with complicated chemistry, but also viruses manage it. Viruses have always managed it. That makes no sense. Then comes another big claim from a paper, which is also completely incorrect. She claims that RNA viruses cannot get through a lipid bilayer, which is partially true. mRNA outside the body would struggle to get through a lipid bilayer on its own. When my students do exactly this experiment with DNA in the lab, we have to use some chemicals and some unusual conditions to get the DNA from outside into the cell. That is because it's comparatively difficult for them to get through. Viruses achieve this by using some kind of proteins on their outside coat that trick the cell into absorbing it typically, or that grab onto sites on the cell, typically proteins or sugar residues, and then use that to inject their genetic material, DNA or RNA, into the cell. It varies what type of virus they are, and it varies what type of cell they're attacking. After this, she jumps onto a completely new topic and says you can look through a microscope and see self-assembly.
We're looking underneath the microscope and seeing the self-assembling nanotechnologies. We're looking under the microscope of people who have been injected and their self-assembling nanotechnologies.
This is possible. You can see some kinds of self-assembly, but not of the kind that she is claiming is happening. So what is supposed to happen here? Self-assembly is when molecules or particles moving around stick to each other in some kind of way that is more ordered than otherwise. Self-assembly can happen with soap molecules on the surface of water. Lipid bilayers can self-assemble in solution, and particles can assemble on the surface of water or on a kind of interface. That can happen. It does happen. But if you're talking about nanoparticles doing something, or nanoscale things, you cannot see it with an optical microscope, and she shows an optical microscope picture, so what that's got to do with anything, I don't know. Typically what you can see in the pictures that she shows are optical artifacts, so there are obvious salt crystals, sodium chloride, those are the cubic things. Sodium chloride crystals you would expect in any body fluid, they happen all the time. They are just a normal feature of body fluids. There should be some kind of chlorides in there.
Why are we being gaslit about a virus that is clinically, statistically, evolutionary, and scientifically impossible to infect people?
So this is claiming that viruses, or this particular virus, cannot infect people, which is, as far as I can tell, based on a study in 2016 about the ancestors of the COVID-19 type of virus, which weren't able to infect people. So it's partially true, which is typical for this kind of thing.
And not talking about the artificial intelligence nano-weapon that's been unleashed on the American people.
She then goes on and talks about nanotechnology, which doesn't really make any sense, because what she's really describing are viruses, which are natural nanotechnology.
And by the way, the spike protein actually matches up with the CAD files, the computer animated design files, in 3 patents for a nano-weapon technology that is made from artificial intelligence magnetic hydrogels.
Now we go on to pretending that it's some kind of nano-weapon without any evidence apart from these images. The images are of a kind of polymer particle, a deliberately made artificial cell, and comparing it to a real cell. So weirdly, they're labeled spike protein, but they're not. These are whole viruses. The spike proteins are the spikes on the outside, which are the proteins, and they're not real images. The corona images are made up. They're computer-generated images. They look like reality, but it's not exactly how they look. And they obviously look like something that has deliberately been generated to be similar to the natural case. These are usually some kind of particles that have been made to fool the body into absorbing them, and so they will look a little bit like cells, because that's the thing that the body is looking for. Likewise, the immune system is most likely to be primed by something that looks somewhat similar to a virus or a bacterium, and so if we want a vaccine to trigger the immune system, it should pretend a little bit to be like it, at least in some ways, which is why there are some kind of patches on the particle, and why it is a gel, so that it behaves otherwise like water, so the rest of it is not interacting with the body at all. We're going to have to divide this claim into several bits. So she's claiming that it's an artificial intelligence, that it's magnetic, and that it's a hydrogel. A hydrogel is just a jelly-like material, like Americans would call jello, like the English people would call jelly, like Germans would call wackelpudding. It is a mostly water mixture containing a polymer, and it can be stiff or relatively soft. Those hydrogels will typically have some kind of function. So in the case of jelly, it's just food, but it contains a lot of water. We could add some chemicals to it to make it salty or have different properties, and that will typically be our hydrogel. There may or may not be anything exciting in there, and it could be made out of mRNA, for example. That would make a gel as well. Magnetic particles are fairly commonly used in biotechnology. They're used in biotechnology because you can collect them again. So if you're using a magnetic particle, if you're using particles to attach to bacteria, because they've got antibodies on, so they attach them, then you can fish the bacteria out of your container by pulling the magnetic particles that are now attached to the bacteria to the side of your container with a magnet. That's why you use magnetic particles. Inside the body, there is no point in using a magnetic particle, because it wouldn't do anything. The magnetism would be distributed over our body, or it would be localized, and it wouldn't really have any effect. What effect would you expect it to have? The stories that she tells of people sticking magnets to themselves are firstly completely debunked. It's quite easy to stick a magnet to yourself. You hide a magnet on the other side of your body, a really strong one, or a piece of metal inside your clothing, and then the magnet will stick. And that is pretty much all you can expect. Anything that makes you magnetic enough to stick particles to would be serious in a different way, because you would be precipitating particles in your body, in your blood, or in your muscles. That would be very unpleasant. Artificial intelligence is another theme. It's a Gish Gallop again. Artificial intelligence is difficult to add to any machine, because it is quite big. If we look at a typical artificial intelligence, so one that I would have used in my research, it's like a desktop PC, maybe 2 desktop PCs, one piled on top of the other. That's about the size of a small artificial intelligence cluster. It would be a high-powered computer, and it would solve a simple problem like recognizing images or reading sentences. If we have a more complicated problem like Siri, and we have a smaller computing power like your phone, then there is a solution to that. Siri is effectively, or the other one, which will be Alexa, is effectively a large supercomputer in a building, taking up several rooms typically, and you talk to it, or your phone talks to it, over the internet with relatively broadband power. What it's doing is it's sending some of your speech to the computer. There's probably some pre-processing. It's sending some data to the artificial intelligence, which works out what you're trying to say, and then sends back the answer. The answer may be, turn on this program in your app list. That's what AI can be used for, but obviously in this case it would be very difficult because it would have to be talking to a tiny chip. On that theme, there's another video on the internet, so I won't show you mine, but a typical dog tag, dog chip, is a piece of glass with a small bit of electronics inside it. It's about the size of a very big grain of rice, like a pudding rice grain, or 4 tiny grains of rice. If you imagine that, the typical needle that's used to inject those under the skin of an animal is scarily large. You would notice if somebody stabbed you with it, it would be very painful indeed. That's the kind of thing that you inject in the dog, and you can feel it from the outside, so that when the vet is trying to discover whether the dog is chipped or not, it can feel its neck and find it in the neck. And then it can scan, and then they can scan it and work out who it belongs to. That's what that chip is for. It has no powered electronics of any kind. All it does is it takes in a radio frequency signal, a high frequency radio frequency signal, and sends back a different one. It's sending back effectively a number which they use to identify the animal. In this case, the AI would have to send some data. She's about to claim later that it's sending some data about your genetics so that you can change it. That would be very difficult because it would have to, first of all, measure whatever your genetic sequencing is, and that requires typically a lab to do. Then it would have to collect the data, compress it somehow, send it over the internet or some way to the AI, which would be located somewhere else, and then it would have to decide and send it back.
So it's not a virus. It's not contagious. How were people getting sick?
They were getting sick with an artificial intelligence nano-weapon.
And how does that spread?
Well, let me explain what it is first.
Yeah.
So if you go back and we go through the research for mRNA, and Professor Catherine Whitehead from Carnegie Mellon, head of chemical engineering, had said, look, we can't do anything with mRNA technology. We can't even do research in vitro outside of the.
So this is an interesting point because she is directly asked why were people dying before the vaccine was invented effectively. I mean, you can imagine that it could have been invented beforehand if you want to be a conspiracy theorist, but that doesn't matter because here you can see that she totally avoids the question and says it only talks about vaccines. What's important about that is that there is no mechanism for this vaccine to get out of the body and get into some other person, although she does say, oh, it's a nanoparticle that can do that, but then it has to be able to copy itself. That's what makes viruses and bacteria dangerous, because they can copy themselves and go into another person. It's exactly the same as the imaginary technovirus from James Bond that she seems to have decided is real. That will be the last James Bond with Daniel Craig, where they have effectively a virus, but it's a nanobot.
What the spike protein is it is this lipid nanoparticle. It's a magnetized lipid nanoparticle. It's called an MPEG that has the CRISPR-Cas9 technology in something called a cubisome.
Now she finally mentions some things that actually are true. CRISPR-Cas9 is an up-and-coming, brilliant way to change genetic code inside a living cell. So you can take a living cell, if you manage to get your proteins and code in there properly, you can change its code while it's running, typically used to change one protein. Of course, in reality, if you were going to put that into a particle to go into a cell to change somebody's DNA. That would be proper DNA alteration, and there is no point to do that for a vaccine. Of course, there is a potential for misusing this technology, but there is no evidence that that is what is inside any virus or vaccine, artificial virus or a vaccine. It would be very difficult to do that currently.
And the AI nano-weapon, what it is, it looks just like the spike protein, and those spikes are actually probes to actually penetrate the cells.
Here she really cuts herself off completely, because at the beginning she was saying that viruses cannot enter cells at all, and now she's saying the spike proteins can allow DNA to, or whatever material, magic material that she's imagining, to enter the cell. And obviously that contradicts her previous point completely.
So they look like cholesterol, and per Ralph Barrick's pitch to DARPA, per the patents, these can be delivered in an aerosol attack. They can be delivered through water. They can be delivered in food. And so what people were infected with was an artificial intelligent parasite.
Here, once again, she brings up the same fellow she was on before, Boric, and he was referring to a natural virus, not to an artificial particle or a vaccine. So this has nothing to do with it. It's completely irrelevant. Okay, here we keep hearing the term AI parasite. We should be asking ourselves at this point, what is the AI supposed to be doing here? If you're going to go through all the effort and expense of hiding technology from everybody, miniaturizing it, and doing something with it, it should have a purpose. There doesn't seem to be any purpose, so it makes no sense. It makes no sense why you would put AI in a thing where a virus would be just as good. A virus is not intelligent at all, but it's pretty good at killing people.
Those spike proteins are parasites. And those parasites do gene editing inside of you to spawn viruses, diseases, biosynthetic structures.
This is an interesting bit, because now we have the spike protein spawning viruses, which previously didn't exist, and it couldn't affect cells. So that is really contradicting all of the material at the beginning.
So when we were told that the vaccine contained the full mRNA sequence for SARS-CoV-2,
that is completely untrue, of course. We were never told that the full sequence for the virus would be put into a vaccine, because that would be extremely dumb. If you put the full sequence of a virus into a vaccine and inserted it into somebody's body, they would start making virus particles immediately, and you would have just given them the virus. That's obviously not what you want to do. If you want to make a vaccine, you make the part of the virus that the body sees and expose it to that, or in the case of older vaccines, you make the virus and kill it, or you make the virus and take the middle part out, the DNA, so it can't copy itself. That has been done in the past. In this case, what they were doing was making the mRNA, which codes for the spike protein only. That cannot make new spike protein, it cannot make new RNA. So once the mRNA from the vaccination has been broken down by the body, which takes a certain amount of time, a couple of weeks maybe, then you can't make any more. Likewise, you can't make any virus particles, because you can't make the rest of the sequence to make any other proteins in the virus, and you can't pack them properly.
Frequencies can also disable this technology, and there is a peer-reviewed publication in Science magazine that actually talks about there are specific electromagnetic sequences that completely disable the spike protein. Why?
This is another part where she's showing a paper and saying that electric frequencies cause something. If we look at this picture, we can see clearly that there is a DC field here. So that is not a frequency.
The 5G towers are part of the energy source for this AI nano-weapon. This towers are taken down, and Starlink was not built for our protection. It's not a military operation for our protection.
Now we're going to 5G. It's another jump, more extra ideas. 5G is a millimeter wave technology. It uses 1 to 10 millimeter wavelength radio waves, end of radio waves. They are usually used for mobile phone technology. The theory here is that they use them to interact with these particles in people. Jumping ahead a little bit, she says something about Starlink. She says it really badly, so it sounds like Starling, but it's Starlink, which she claims is a military endeavor. It's not a military endeavor at all. It's a private endeavor by Elon Musk, and it's something to do also with internet. The wavelength of that is a bit longer. It's about, I think, 20 millimeters, 23, I think it is. So that's the kind of wavelength we're looking at. To be able to interact with that kind of light, electromagnetic wavelengths, we would have to have an antenna of somewhere around that size. It doesn't have to be exact. It can be a little bit smaller, but it would have to be somewhere around that size. Obviously, these nanoparticles or even microparticles are way too small to be able to interact with energy at 5G frequency. If we go up to higher frequency to be able to interact with the body, then we have the problem that we're going to interact with a whole lot of other things, so our penetration won't be very good at all. We won't be able to get to all the people. That's why that wavelength has been chosen. It's a wavelength that it doesn't absorb very well. There isn't a very easy wavelength to be able to excite nanoparticles and get everybody. So it isn't true.
Very early on, probably a year and a half ago, I was looking at images on this show and I said, that looks like a self-aware organism. People looked at me like I was absolutely insane.
Yes, it is. And it says in all, so magnetic hydrogels, genetically engineered magnetic hydrogels are self-aware.
Okay, so here we've got another thing. We've now started to claim, or they have now started to claim, that these particles, or whatever they are somehow self-aware. That is fairly extreme. Something becomes self-aware when it is able to think beyond a certain amount. A bacterium is not self-aware. And a magnetic hydrogel is definitely not self-aware. It is magnetic and it's a gel. It can move in a magnetic field, but it isn't alive. It can solve a maze maybe if you set up the maze very, very carefully to be solvable by a magnetic blob of glue, but it doesn't think while it's doing it. They both claim that it's self-aware, and I suspect this comes from the phrase smart materials. People have been using this for a while, and you call things a smart material when it has extra functionality, usually can sense something. So if you take a magnetic particle, it's a smart material because it behaves as a particle, and then when you apply a magnet, it does something else. It moves in the magnetic field to where you want it to be. A hydrogel could be a smart material because if you put a drug in it, as the hydrogel breaks down, it releases the drug over time. That's smart. The thing that we're thinking of as smart here is not the same as self-aware smart or as AAI smart. The idea is a sales thing. Us material scientists are guilty in this case of trying to sell our work as being clever, in which case we've made a drop of liquid which will move in a magnetic field because it contains some magnetic particles, or we've made a material that is full of glue blobs so that when you snap it or when you crack it, the glue runs into the crack and sticks it back together again. That is a smart material. The level of smart is way less than they are imagining here. So there you are, that's all of their points. Please remember that it's important to remain critical and think about things carefully, even the ones that you agree with, so not them, me. You should still be critical of things that I say, but analyze and think about what kind of dirty tricks that they're playing. In this case, they were quote mining They were Gish Galloping, and they were jumping themes. They were using... So I've got to the end now of their points. There's a bit more video, but there aren't any new points. They just go over the same things over and over again to try to beat it in. They don't come up with any new evidence. And there isn't really any evidence at all for their main points. The only evidence is only tangibly related and is missold as being evidence for something that it really isn't evidence for. So what does that mean for the future, or what is my main message here, is there are some bad tricks that people use, and it is good to be able to notice those so that you can understand how they're trying to manipulate you. Maybe they've been manipulated themselves and don't even realize it. It doesn't mean that you should accept everything that an official tells you. That is not what I'm trying to tell you. The official line is very often also incorrect. However, the same thing applies. There are usually some fairly obvious signs of how they're trying to hide their misinformation, and if they have data, you can usually look at it and find out what is really behind it. The example here is the more recent thing that came out of Pfizer saying they hadn't tested their vaccine against transmissibility, which is a thing that we really knew all along. Vaccines are never tested against transmissibility. It's fairly normal to assume that they will reduce transmissibility, but it is difficult to test for it, so they're not tested. It is not a massive victory to find out that they weren't tested for something that they never are tested for. However, obviously, a lot of people in the health industry, and politicians mostly, missold quite obviously and probably very badly their products and their ideas more. It was more people in the push industry of trying to get everybody to do something that they wanted us to do, oversold it. And that is also a good thing to be able to see when they're doing it to you.